Good afternoon. My name is Fernando Cruz. I work in the CNAG as a data analyst in the genome assembly and annotation team. I entitled this presentation as Bioinformatic HiC, a bridge between the library preparation and commercial level assembly. So I divided in four main blocks the talk. One will be an introduction about the CNAG, the HiC. A second one about how to read the bioinformatic QC, and particularly focusing on the approach we follow, the pipeline, and mainly based on the dovetail preprocessing. And then how we use the HiC reads for obtaining chromosome level assemblies, and in the end some conclusions. So we are based in Catalonia, in Barcelona, so a bit far from Sweden. And basically the CNAG was created in 2010 by the central government of Spain and the local government of Catalonia to create an infrastructure for sequencing and enhanced genomics project. And we are nowadays more than 100 people working, and it's still growing. And it's directed by Ivo Wood. And so the kind of the mission or motto of the CNAG is try to improve people's health and quality life by using genomic analysis. So we aim at being a high quality center for sequencing and also complement this sequencing with genome analysis, highly specialized. Okay. In fact, we collaborate with different universities and research centers, and we provide free bioinformatics analysis, and there is kind of a verbal agreement that the data analysts and the bioinformaticians will be co-authors depending on the contribution to the research paper. Okay, so it's kind of staff scientist work somehow. So the sequencing capacity of the CNAG is quite, I think, is quite high. So we can sequence more than 10,000 gigabase of human genome in one day. We have a wide variety of Illumina sequencing instruments and a certification, quality certification for Illumina. Another one for nanopore, and nanopore has arrived very early to the CNAG, so we have from the little mean ions to the green ion, and now a atomic ion with 24 cell process. And then we have also single cell genomics, the 10x chromium, and some people do that special genomics. Honestly, I don't know how they do this. I'm not very familiar with this technology. And then we have a very good computing capacity. So we have plenty of space and CPUs and cores and nodes to compute the assembly work and the mappings. And actually, the new nodes we have, all of them, they have 1 terabyte of RAM, and a few of them even 2 terabytes of RAM. So we can deal with nasty genomes and be able to conduct the reads. So these are the strategic areas. I will focus just in 2 of them that use HiC. So beside the rare diseases and cancer and single cell studies and personalized medicine, we have the genome in action that is mainly led by Mark Marti, that's 3D genomics. And then this is biodiversity blog, where is the genome assembly and annotation team, and the BGE project, the one that funded this course, is the one that we are mainly working on, besides also the Catalan Biogenome project. So this funding from the local government, and also to produce reference sequences for the EBP. Okay. So that's the global view. The units involved are under the Marta Wood is the head of sequencing, and we work in close collaboration with the lab and the genome assembly team led by Tyler Eliotto. Laura, that is here leads now the long-read sequencing team, and me or Jessica or Francisco, we run the QCs when we obtain the data for shallow sequencing, from shallow sequencing of the libraries. So, the Bioinformatic HiC QC, all we know this protocol, okay? So there are some crucial steps. I think it will be the cross-linking, especially. All of them seem to be crucial in the end, and the ligation also is very important. But in the end, what the aim is to have a pair of sequencing reads that comes from a hybrid molecule that contains two different parts of the genome, let's say two different chromosomes, that they are attached together and they were in close proximity, in contact in the nucleus. Okay? The protocol, as you have seen, can take minimum 3 days and 14 days. Laura told me this before we started, but we already confirmed that minimum you need 3 days to do a HiC library. And on top of that, you have to add a little bit of time, maybe 1 week, to prepare the sequencing library and then run some shallow sequencing if you want, or go for deep sequencing if you are in a hurry. But everything is okay. But when we work with challenging animals, things start to be more complicated, and the times are actually close to these 14 days or 10 days, or you have to repeat the libraries one and again. So the basic idea is that we have to link the genome, then we digested it, and then we will have some fragments that will be sequenced, we would not like to consider, and some fragments that are really informative. This is in the HiC ligation. Okay, so this is in the jargon of HiCap would be a valid die tag. That would be a read pair that comes from a hybrid or chimeric fragment from the genome that has a restriction site where it was like the ligation occurred. Okay, and then we will have things that comes from fragments in close proximity, either consecutively or the same fragment usually ligated. Okay, so these cases will contribute especially to short cis contacts below 1 kb, and these others to contacts larger than 1 kb in cis. Cis means that you map the reads to the same sequence block, let's say a contig or a scaffold. And then you can measure actually the distance between the alignment of the first read to the alignment of the second read from the same pair. Okay, so then you have an estimation of how far away these sequences are in the genome. And trans would be cases where you map one read to one scaffold or contig and the other read to a different scaffold or contig. Those are really good for us for scaffolding, because I also have to join these two scaffolds or contigs into one super scaffold or genome territory. Okay, so then there are several HiC QC and read preprocessing pipelines, because we're interested in doing the QC, but also in preprocessing the alignments to be able to use them for scaffolding. So all of them are based in mapping, and in the case of these three, the HiC pipeline, the OmniC, and the user, they are mapping based, regardless of the use of residual enzyme or DNAs. Actually, you can run them in a mode where you don't specify the restriction enzyme, the restriction site. While with HiCap, you explicitly need to specify the cut sites. Okay, so each company is used to a different set of stats and have their internal records that can compare against our results. So to produce ARIMA-friendly reports, my favorite is to use HiCap. I found that very easy to reproduce and to install the content environments for different versions. And HiCap is based in the fact that it recognizes the restriction site both in the reads before mapping. So whenever it sees the junction, the restriction site, it chops the reads and discards the rest of the read. You skip the 5' end. And then these reads, truncated reads, are mapped to in silico digested genome, where you use the information about the restriction sites, and you, whenever you find them in your assembly, you just chop the sequence, and you obtain a digested reference. For the dovetails pipeline, here's just an overview of the results. But the basic idea here is, I think it's more resilient to the fact that you have restriction sites or not, or adapters, because basically you process the alignments and you aim to have the best 5' end alignment of each read from the same pair. Okay? And you select the best unique alignment. We'll go over again the same thing. In this case, you will have a chimeric read that HiCap will truncate, but the dovetail pipeline using per tools, different program, basically we will select the best 5' alignments. Okay. In this case, will be a trans alignment with the red alignment mapping to one chromosome and the blue to a different chromosome. Okay, so here I made a little comparison of a library from this butterfly, Revia palarica, where we produce an ARIMA library with, and we sequence 1.7 million pairs. When I said pairs, read pairs, but let's call it pairs. So the numbers vary a little bit slightly between both pipelines due to different reasons, mapping thresholds, etc. What you have seen is that the global picture normally is very similar. Like in this case, we have 30% sort Cs. In this case, we have 33.89%. And what I also like from the dovetail pipeline for scaffolding is that we seem to rescue more valid pairs. In this case, we rescued 633,000, and in this case, 580.59. Well, 599. Okay, so that's it. So I have some examples where bad libraries are basically bad in our report using HiCap and a report using the other QC pipeline, the dovetail pipeline. Okay? But in terms of also preprocessing the alignments to use the data for scaffolding, we decided to integrate the dovetail protocol in our SnakeMe pipeline called CLOS. It's an assembly pipeline that we're using for this project in the CNAG and automatizes a lot the tasks. So there's a mode. This is a part or a section of the pipeline with a flow chart summarizing the steps required to do the QC using the dovetail pipeline. Okay, and with a few complementary steps. The first step will be aligning with BWMM both grids at the same time using certain mapping threshold. Normally you use 40, that is the default recommended. That means that it's kind of the threshold for uniqueness of your alignments. And it's in logarithmic scale, like the Fred score. So in this case, you will have 1 over 10,000 alignments that could be ambiguous. If you have a mapping quality of 50, you will have 1 over 100,000 alignments that could be ambiguous. So this is kind of our threshold for uniqueness of the mappings. And we not regularly do running with 40 and 0 to tolerate multi-mappings, and in some cases with a range of mappings. Then the pair tools is a package with different programs, but basically what it's going to do is record the valid ligation events. And we remove the PCR duplicates and keep those paired mappings that are unique, what is called no dupe in the Dovetail jargon. In complementing this, we also take a subset of 100 map reads, and we do a read screen that is basically blast them against NT. Why we do that? Because at some point we have very bad libraries with sponges and stuff like that, and we had a lot of map reads, and we didn't know if these reads were the reason we have that many map reads was because we had a contaminant competing with our reads, and we couldn't map the reference from some other reason, like there's not enough amount of cells in the list or something. So that's why we regularly do this as a complement. And then after we have the alignments in this BAM file, we run the QC statistics that allows us to classify the read pairs and determine also the library complexity with certain accuracy, especially if the number of reads between 1 and 2 million. Okay. What happened? That normally it's very hard to fine-tune the number of million reads due to the huge variation in the size, as in the libraries where you can have small fragments and fragments up to 15 KB, maybe, in your library. And then to estimate the molarity and run it in the NovaSeq seems to be a problem. So we see a huge variation. Something that I should have thought before, but we can fix this just downsampling the number of reads we have when we have too many. So one of the first assemblies we have released in the BGE project is the Rupi-Capra. Rupi-Capra is a sample coming from Slovenia, and it's already uploaded to the ENA, also got the seal of approval for the sequencing assembly committee, and we got a chromosome level where we have 27 autosomes and 2 sex chromosomes. Nice assembly. By the time we had the, I think it was OmniC library. By the time we produced our OmniC library for QC, we didn't have a reference yet. The anti-reads were not ready. So we decided to use the domestic goat chromosome level assembly. That was one of the first domestic animals to have a chromosome level assembly. And the divergence time is relatively short, 6 million years, like human and chimp, 99% identical in sequence in principle. So ideally, you should have the same species, but if not, at least aim for an assembly that has a contig 50 above 1 megabase, and it's it comes from a close species. So this is the core of the Dovetail QC stats. This is table. It's around this table, and then you have a pair file also that you can use. So I will go step by step, explaining the different stats. Are very similar to what you get with the HiC, slightly different. The thresholds are different for the short Cs, for example. Here, what is alarming is the Cs below 1 KB. While in HiC you have short Cs below 10 KB, long above 10 kb. But yeah, the results are very similar. So then the first block of statistics are basically proportions of the total number of pairs sequence of raw reads. Okay? So this 7.6% comes from dividing this number by this 3.5 million reads. Okay? The same with the map reads, the PCR duplicates, and the no duplicate pairs. So, just translating these two images, although I need to improve these plots, you have this, the total will be the total number of reads sequence, and these three, the map, the low mapping quality, and the map, will add up to 100. Okay? So, the map is whatever was a map to the genome. That means that maybe you could have map one read, but not the other. So it's a map read pairs. The low mapping quality is what we consider multi-mappings, because are below our mapping quality threshold. And what we consider maps is every alignment of the two reads that is about this mapping quality threshold, that if you remember right, is 40 most of the time. And then you have a percentage of PCR duplicates and a percentage of no duplicate pairs that are a split of this 65% of mapped read pairs. Okay? So then, the total pairs depends on the sequencing depth, basically. Although in the shallow sequencing, you can be affected by the variance in fragment size in the library. The map reads also depends on the divergence to the reference sequence, the possibility of having contaminants in your sample. The map reads are those that both 5' ends are fairly well mapped. Depends on the mapping quality threshold you use, the repetitiveness of the genome. So for repetitive genomes, we tend to drop it a little bit to be sure that we recover more pairs, and also the sequence divergence to the reference. Then we have the low mapping quality reads that are the opposite of the mapped ones, are those below the threshold. Depends on basically the same things that affect to the mapping. And then we have the PCR duplicates. This category is basically when we map reads that have exactly the same start and end coordinates. That couldn't happen by chance with a whole genome Saturn library. So those are indicative that in the PCR may have ended up having multiple copies respect to other fragments. Okay? Comes from the same fragment and was sequenced over and over. But also depends on the number of cycles you use during the digestion. Well, actually, it's not during the digestion, it's during the library prep for Illumina, no? So this is wrong. And also, it's affected by the sequencing instrument you use. The recommendation, both for OmniC and Dovetail, ARIMA and Dovetail, I think is use MySeq. That could be more precise and has a lower rate of duplicates. But we tend to go a step forward in the next step, NovaSeq 6000, that we know that has a basal optical duplicate rate due to luminical contamination between clusters of reads, okay, in the flow cell. But anyway, we know that, but we still think that when this number is high, it's mainly due to problems during the HiC library preparation that have increased the amount of PCR duplicates. Okay. And in the end, we have this category that is the one we would actually deal with, is the unique mappings at both 5' ends. And basically depends on the factors that affect the mapping, and competes with the PCR duplicates for the pairs. Okay, so you don't want to have many PCR duplicates. You would like to have many no duplicate pairs. So then we go to the second part of the table. The second part of the table, they change the total. Okay, so everything is basically focusing on the filter rate. So it's a proportion of the non-duplicate pairs. Okay? So then you have the cis, the trans pairs, the valid, that would be all cis contacts that are long enough, at least longer than 1 kb, and the trans. And then you have these very short pairs in cis that map short distance shorter than 1 kb in the optical scaffold. Then you have the cis intermediate lengths and the cis long lengths over 10 kb. Okay. So what we know? We know that there's an inverse relationship between the proportion of trans pairs and the contiguity of the genome. So normally we expect that with when we have a high chromosome level assembly, we will have a higher proportion of cis and a lower proportion of trans. That's what we see in this example. Before scaffolding, we have 40% cis, 60% trans, and the opposite after scaffolding. Normally they revert. The cis is clear. I'm referring to pairs that map in the same chromosome, are useful to confirm the order and detect misjoins in one chromosomal or super scaffold. And the trans will be those that are very useful for scaffolding because they map to different chromosomes and they will always, well, with very good assemblies, when you map the HiC reads, you easily localize chromosomal territories with sometimes even without the scaffolding. So, of course, the bioinformatic QC is totally dependent on the reference. You need a reference, otherwise you cannot map the reads. You have no contiguity in your assembly. You will expect to have a lot of trans mappings, a huge amount of trans mappings. And then you said, okay, it's not that bad library. Because I have an assembly that has an M50 of 500 kb, and then I have 80% trans mappings, trans pairs. Well, that's okay. Unless you have another stat that is telling you that something fishy happened during the HiC. And then when you have your genome in chromosome level assembly, you would expect to have most of your read pairs in cis and not in. So I normally plot also these stats, and I also calculate kind of a made up stat that is the sequencing efficiency. Because I was seeing that we were sequencing a lot of pairs, but in the end, few of them were valid. Okay? So then I wanted to know the proportion of valid pairs versus the total number of read pairs that we have sequenced. So here, for example, we have a sequencing efficiency in this relatively good library, RuPiCapra, of 48.9%. Okay? Not bad. Then what we have that is nice is like we have almost 60% cis pairs, 40% trans, and 80% valid pairs. Okay? By valid are these two categories: the cis longer than 1 kb and the cis longer than 1 and longer than 10 kb. Basically, longer than. Plus the trans. So you add up the green chart and the violet chart. We have short contacts, short cis contacts that are very, very close to each other. That presumably come from a fragment that is very short, and the distance between them is below 1 kb. Okay, so the red flags are PCR duplicates for the mapping, and for the filter reads the short cis contacts. Okay, so what I really like to do also is what the doctor does, and also I think the Sanger does in a different way, but basically you plot the distribution of your cis contacts in y-axis, in the y-axis is the frequency, and the distance between the two pairs in cis is in the x-axis in log 10 scale. So 3 means that is 1,000 base pairs length. Okay. So what we normally do, you see, is for a good library, is a bimodal distribution where you have a peak of short chunk below 1 kb, and then a decent amount of pairs that are up to even 1 megabase, 10 megabases, or 100 megabases in distance. Take into account that to do a proper estimation, you need to have a highly contiguous reference to do this. It will be slightly constrained by the contiguity of the reference. But even though, if you see an excess of this kind of reads, it's bad. So, next slide is actually an example of 2 different libraries for the same species, where we see this very bad library where most of our cis contacts are below 1 kb, and almost no pairs are in distances above 1 kb. Okay? This was our good library, this is our bad library. And you see this, or it doesn't matter the protocol you ARIMA or OMC, you see this every now and then. Depends on the sample, or who knows. So another statistic that Dr. used, but I realized that we haven't been using it. I didn't pay much attention to it because I normally use all the reads I have for the shallow sequencing. What I should be using 1.5 million reads or something like that, to be more precise, is the library complexity. That is, in my own words, it would be like the variety of fragments from different parts of the genome that is contained in your library. You want to keep it principally below 300 million pairs, and a relatively good library, okay to go, will be almost 100 million. And for a good library, you will have 220, 250 million read pairs in this cell. Okay. After talking with different people and looking at the results, kind of summarize what would be good thresholds or ideal values for the different stats. The map reads, you would like to keep them below 10%. The map reads at least over 80%. The PCR duplicates below 6%, but the higher it is, actually, the higher is the number of PCR duplicates when we do the deep sequencing. And then you would like to have at least 60% of read pairs mapped uniquely. And then, let's say 40% is below 30% trans, above 80% cis, valid, sorry, about 80% valid read pairs. This is because I would like to have more or less 60% of the, for a good library, for a good human library, what I heard from my colleagues is that normally you expect to have like 60% valid pairs out of the total read pair sequence, no? Then you would like to keep this number the lower as possible, and values above 20% are not any good. These are the C-sort contacts below 1 kb, okay? And this, the Cs above 1 kb, you would like to have at least 30% and at least 20% of the long range cis contacts. And as I said before, between 100 and 250 million reads of library complexity should be a good value. So once we have done the QC, we give green light, we discuss among the lab and observe, and we say, okay, let's sequence the library more deeply. So then, what I'm interested why I'm interested in this data? Because we are doing genome assemblies. And even if you have a really high contiguity, really nice assembly, to start with just nanopore reads, that is basically what we use. We've been using BioClear reads, and we expect to use the Revio Hi-Fi reads soon, support to arrive this summer. So even if you have a very good assembly to start with, you will need to make some corrections and verify that the chromosome supercouples are correct, and everything is in the right place. So the sequencing recipe that we are using in the BGE project at CNAG, at least I think others might be using a similar one, is we use Illumina 60X because we want to polish the assemblies done with ONT. Although with Q20 now we are getting really nice consensus qualities for quality values. ONT above 60X, and a HiC library above 50X increase. And then we use all this data to produce a HiC scaffolded assembly. Basically, we preprocess the reads, Illumina with Finvalore, ONT with Finlong. We assemble the reads. Normally, we use two different assemblers, Fly and Next de Novo. And then we use the Illumina reads plus the ONT to perform normally one round of polishing with HiC. Okay, so in this first part, we don't use the HiC. Then we push haplotics. Basically, we have contigs that belong to one haplotype, let's say the maternal haplotype, and another contig that aligns quite well with this one that belongs to the father chromosome. Okay, so then we push one of them, normally the shortest, and we keep it in the assembly. Then we avoid having artificial duplicates in our assembly, like false paralogs or stuff like that. And in the end, we drop a grain of salt with the HiC data. And we do the HiC scaffolding. So then, as you see here, we have this contact map before scaffolding, where we have some little contigs that, okay, these ones should be dragged here and belong to this chromosome. And the same here, this one is likely belongs here. Okay. So then, this allows us not just to do the scaffolding, but after the scaffolding, we need to do the curation. We need to detect falsely misjoined scaffolds. In this case, you see a sharp contrast between the one, I would call it chromosomal territory, and another one. So we should break this scaffold here, or this contig here, and have independent contigs. Then you can detect misjoins. There are things that, come on, this is not belonging to this particular place in the genome. This seems to be coming from here. No, this was a misjoin, this is a translocation. Okay. This belongs here. So we move it and we place it in the right place. And then you also end up having these X patterns that what they are telling you that this sequence contig is inverted. Because this part at the end of the contig actually have stronger contacts with the end other scaffold. Okay? And the beginning of this one with the beginning of the next scaffold. Okay, so in this case, this is basically a continuation. You go and you fix the inversion. You flip the orientation of this particular context, and everything has a nice, the contact map looks much nicer. You have a strong diagonal signal with a checkerboard pattern, but everything, the contacts correlate with distance, the linear distance in the chromosome or in the superscaffold. Okay? At some point, we will ask that. But how many HiC data you need? How many pairs you need to do a good chromosome level scaffolding? Blah, blah, blah. So that got me thinking, okay, what people do, no? How many contacts we would need for scaffolding and curation? So there's nothing written about it. So, hey, one of the first articles that came out was kind of groundbreaking. They were aiming at sequencing vertebrate genomes, well, mammalian genomes for cheap, for under $1,000. This is a paper by Olga Duchenko. The recipe was to prepare an in situ HiC library with 100 million reads. And then, assuming that a good library will have at least 60% of valid read pairs, then that means that I could estimate kind of my HiC density. That would be the million valid pairs per gigabase of genome. For this case, the Aiden lab with 100 million pairs will end up having 20 million valid pairs per gigabase. I think that's too little. But the recipe of the BGP or the Darwin Tree of Life or even the ERGA, well, the ERGA is 50x, well, BGE is 50x. Let's say that you will end up for a 3 gigabase genome with 600 million pairs. If you make the numbers for 150 base per read, you get this. So that will imply that we will have a HiC density of 120 million valid pairs per gigabase. Something in between these two values is a reasonable amount, in my experience. So for Rupi Capra, we got over 60 million valid pairs per gigabase of genome. Before scaffolding, we have 64.76. Okay, let's see what happens when you have low HiC density. These are two vertebrate genomes, totally anonymous, that have more or less the similar genome length, 2.2 and 2.3 gigabyte bases. The one on the left has a HiC density that is 30 million valid pairs per gigabase of genome. And the one on the right has a density of 120 million per gigabase of genome. This one looks really nice. And this is after scaffolding already, but you see the chromosomal territories are nicely defined and actually require minimal curations, this genome. In this other case, we already have, in the first super scaffold, a case where we have a 563 megabase super scaffold. That is more than double the size of the largest chromosome for this species. So this is bad. Of course, we can create this. We can look at alignments with close species and also say, come on, this is, seems that here is a sharp reduction in contacts between this part and this other. Maybe we should click them. Actually, this super scaffold was split in 3 or 4 smaller scaffolds. Okay, so then our relatively good example, Rupi Capra, had 64.8 million valid pairs. At the beginning, we had an input M50 of 78 megabases and a total number of scaffolds of 110. After scaffolding, we ended up having scaffolding and curation. We ended up having 30 super scaffolds, okay? So this is consistent with the karyotype. It's like you have 28 autosomes plus 2 sex chromosomes, because this was the heterogametic sex, this was a male. This is very nice. It's very nice, and also allow us to detect both and identify both due to this high C coverage around the chromosomal superscaffolds and the ONT coverage or OV coverage. We can detect the sex chromosomes. Okay, we see that this one, the X chromosome, has half the coverage than the autosomes for the ONT, and the same you see in the depleting of contacts of the diagonal for the high C. So both data are consistent. The same happened with the chromosome Y, although the chromosome Y contains a lot of repetitive sequence that explains the peaks in the coverage. Okay? So it's nice that you can also identify the sex chromosomes in cases where you know the karyotype and you are sure that you are sequencing and assembling the heterogametic sex. This is an example where we had a very bad library for a butterfly genome. And we ended up having 3 million valid pairs per gigabyte of genome. The genome is below, is half a gig, but I converted it to pairs by gigabytes to be able to compare. And in this case, we don't, luckily, the quantic assembly was so good. The genome size was small and the quantic assembly was so good. The M50 was 24 megabases that we already have everything almost into chromosomes. Okay? In a total of 25 scaffolds. We just needed to do what we were able to do to fix one translocation and one misjoin. And we ended up with a chromosome level assembly that was 90%, 99% complete with a low duplicate rate. And now it's accessible in the ENA. This is good, but it's not always the case. You can have a mixture of low contiguity plus low high density. But let's be optimistic and think that one thing can always complement the other and try to get a good genome reference. Okay, and the high data is also important for kind of quality control of the assemblies, because the sequencing and assembly committee have put in place this method where we compile different assembly metrics into a single report, and then we share this report together with a link to the contact map with another expert in the field to review our assembly. And then this way, we have an independent evaluation of how good our assembly is. And someone can tell, look, Fernando, maybe these short scaffolds you have at the end, contact with this super scaffold at the beginning, move them there, try to okay, you localize them there, try to place them inside if you can or not, resolve some haplotype, things like that. And once the revision is done, we send this report, they evaluate the contact map, we send the metrics. These metrics are above the EVP standard. We get a seal of approval for our assembly to be uploaded to ENA. And that's all. But I would like to just comment a couple of things. So I think the bioinformatic QC is important because detects problems that sometimes are invisible to the lab. Because all the metrics, the QC1, for example, metadata can be good, and in the end, we end up having a library that we have a lot of Cs contact. That happened to us. The same with only C. we see nice digestion profiles, and then in the end, same story, very short cis contacts. They are essential to obtain high-quality chromosome-level references, but still, the challenging organisms and samples, we are learning with them, both to assemble and both to prepare the HiC libraries. And often we don't know exactly what's the enemy, because we don't know what's the problem. We need to figure out which is our enemy, okay? Thanks for the attention, and feel free to make some questions and comments.